instrument is supplied in a safety case. This is how the setup of the Aquaflux instrument will look like. Connect the power cord to the back of the base unit. Connect the PC with the base unit via USB. Connect the probe cable to the front of the base unit. Switch on and boot up PC. Start the Aquaflux program. When requested, enter the probe serial number. On the base unit, switch to measurement mode. Base unit light turns blue. Pick up the probe and touch the condenser at the back of the measurement chamber repeatedly with the stem, not the sock tip, of a clean sock-tip swab. Use light scribing motions for about 10 seconds. Replace the measurement cap with the calibration cap and park the probe on the base unit. You can see in the flux density monitor tab that the graph is nearing zero, indicating there is no flux. Wait for at least 30 minutes until the flux readings have settled to a steady level. In the Aquaflux program, select the Baseline Calibration tab of the control panel and click on Calibrate. In the Aquaflux program, select the TWL tab of the control panel and click on the button with 3 dots to enter a file name for today's measurements. If you forget, then the software will create a default output file. No data will be lost. You are now ready to take TWL measurements. Replace the calibration cap with the measurement cap. If necessary, fit a different measurement cap and adjust the cap factor accordingly. For in vivo measurements, i.e., measurement of natural human skin, standard measurement cap or other caps can be used depending on the location of the site to be investigated. Place the probe onto the skin surface at 90 degrees with adequate contact pressure so that there is no gap between the probe and the skin site being investigated. And place one finger onto the red button on the probe. Press and release the red button. You can see in the software that the measurement tab is automatically opened and we can see a graph being plotted indicating the flux value. As the flux is being after, at some point the graph starts becoming flat and we can see a horizontal bar appears. This bar is an indication of the expected RST and the current RST. When the flux levels are settled, the RST comes close to the expected value and the measurement stops automatically. Once the flux reading graph stops, the button at the bottom right of the software changes from a board to start, indicating that the instrument is ready to take next reading. You can either place the probe in the stand in sealed position or can directly move onto to the next site. According to FDA, no more than approximately 15 grams of water per square meter per hour, i.e., less than or equal to 15 gm to HR, could be a reasonable skin barrier integrity acceptance cutoff criterion for a TWL barrier integrity test on human torso or thigh skin. Once you have completed taking the readings, you can shut down the instrument and PC by following these steps. This is the Sensors tab where we can see the current, ambient humidity, and temperature of the surrounding. We can see in the temperature graph that the temperature of the condenser plate is around -5, indicating that there is ice formation at the condenser plate. At the top right corner, there is continuous display of the flux value, and the surrounding temperature and humidity are also displayed. The ideal conditions at which the instrument should be operated are that the ambient temperature should be between 20 to 25 degrees Celsius and relative humidity should be less than 60%, lower the better. Below the ambient conditions tab, we can see the ice gauge, which is an indication of the ice accumulated in the measurement chamber. Once the gauge color turns orange, we need to switch the mode to standby mode and keep the probe in the non-sealed position to allow the accumulated ice to dissipate. This is the flux density monitor tab screen. This is the measurement tab. This is the results tab, where we can see the details of each reading and add comments for each reading also. If one wants to view the data in the form of curves, then one can select the Flux Curves button in the Results tab. If one wants to view the data in the form of histograms, then one can select the TEWL Histogram button in the Results tab. There are different caps available for different applications. Here in this video we'll show two examples of caps and adapter. For the in vitro measurements, i.e., for measuring TEWL of excised skin samples, the standard measurement cap might not be suitable, so the cap is changed. Whenever a cap other than the standard measurement cap is used, the cap factor value in the software should also be changed before taking the readings. This cap is used for Dermax small cell cap of orifice 11.3 mm. Just remove the standard measurement and replace it with this cap. Then you can just place the probe on the acclimatized skin sample and start the TEWL measurements. The excised skin sample should be well acclimatized before taking TEWL readings. Follow the same procedure for the standard France diffusion cell. Just remove the standard measurement and replace it with an adapter that can be used for France diffusion cell.